Thank you very much for this invitation. It's a pleasure to be here. For me, it's the first time in this center, which is well known all over the world, but not in the campus nearby. But yeah, coming back here. So I selected a title which is a little bit speculative, The Power of Networking, but I would like to show you that we have a plant microbiome which really interact with each other. So I have completely the opposite structure than Philip. So I will start with the microbiome and then go more into the detail. And the most important thing is health, plant health, but also human health. But I will only more focus on plant health. So this is our view on all eukaryotic organisms now. So we know that all of them contain a microbiome, a specific microbiome, and a lot of interplay and interaction with the host in many important functions. So we know it for a long time. Here are several colleagues which I know from the Rhizosphere conference, and more than 100 years it is known that the rhizosphere microorganisms have these important function for the plant host, but recently also the people from human medical microbiology realized that everything is connected and we really have to see all the eukaryotic hosts as a holobiont. And for sure we want to avoid any pathogens or outbreaks, and in this case it's very important to study the microbiome and understand the balance of the microbiome. So, and as I told you, we have these networks, or the microbial networks or microbiomes, and first I would like to show you several examples of our projects. And then I will show you how we can use or exploit this for health issues. So I have not so much time, but I selected 5 different model plants and crops to give you an overview about our projects, but also that you can get an idea how specific these microbiomes are and which functions they can fulfill on their plant hosts. So I will start with 2 model plants. The first are mosses, then I will show you some results of an Arabidopsis project. Then I come to medicinal plants, which are also very specific. So we have learned in the talk of Philip that there is a difference between the monocots and the dicots, and especially the medicinal plants, so they have a very rich secondary metabolism. Then an important vegetable and at least crop. So the mosses, I start with these investigations when I was in Germany. In northern Germany, we have a huge territorium covered with bogs ecosystem, which are very important for our world climate. And when we started, there was more or less nothing known about the moss microbiome, but we thought it's very interesting to study them because they were the phylogenetically oldest land plants, and maybe they have a very intimate interaction with their host. And if you look on bog ecosystems, so these are more or less monocultures, but they have normally they are very healthy and stable over centuries. And it is also known that they have a high antifungal activity, so they were used in traditional medicine, but also as baby nappies here in Northern Europe, and also as wound coverings during World War II. So only some of what I think are the highlights of these projects, which we had for a long time. So here, several years before, we sequenced the first metagenome of a moss microbiome. So at this time there were not so many metagenomes available. So here you sequence the whole genes of the microbial community, and you can see here in this fish picture that there are a lot of endophytic bacteria inside of the moss. You can visualize them, and they have really a high number and amount. So at the end, then our metagenome was in between the plants and freshwater, which fits well. And then it's also possible to go into detail and to compare your metagenome with all the other metagenomes which were available. So, and before we sequenced this metagenome really in depth, so we developed some hypotheses, and one was for sure that the microbiome protects the moss plants against diseases, and also protects them against abiotic stress, because the life in the bog ecosystem is not so easy, so there are a lot of abiotic stresses which they have to cope with, and they have also a limited amount of nutrients, because many of them live ombotrophically, which means that they only live from the rain. So, and therefore we also looked especially on these points, and we could here confirm our hypothesis, because there were much more genes involved in stress tolerance than in other microbiomes and in nutrient supply. And what we have not expected was that there's so many communication within these bog ecosystems. So I saw that they are more slow and that everything is in the balance in the ecosystem, but it seems that they communicate and exchange a lot to survive under these conditions. So coming to the network, so it's also possible then to look on these networks which the microbes form. And here we compare two different functions. You can see the nitrogen fixers on one hand, and here below you can see the metanotrophic genes. We compared two Sphagnum species, Sphagnum phallax and Sphagnum magellanicum, and what was a surprise was that the nitrogen fixers, they form a very specific network. So phallax has specific nitrogen fixers, for example, from the Burkholderia genus, or Magellanicum from Methylobacterium. So they were very, very specific. And on the other side, these metanotrophs, they were shared by both mosses. So, and our explanation was that the nitrogen fixers really belong to the plant and have a function for the host plant. In the opposite, these metanogens, they are important for the whole ecosystem, and therefore they share them. So our result was confirmed also by our study between we studied the transfer from the gametophyte to the sporophyte, so to the next generation of the moss, and the capsules, the sporophytes, contain the spores, but they also contain a core microbiome, which is essential because most of the mosses, they cannot germinate without their bacteria. And interestingly, a lot of nitrogen fixers, or the majority of nitrogen fixers, was inside of these capsules, but not the metanotrophs, which they acquire from the environment. So, next project, this is an Arabidopsis project. This was done in cooperation with the Max Planck Institute in Golm, and for a long time they studied cold acclimation, which is an important procedure and restricted our plants to a specific geographical area. And it it is interesting to study this by using an ecotype selection of Arabidopsis. So this is a natural connection, well studied. And you can explain these different cold acclimation of the ecotypes by several metabolomic data of the Arabidopsis plants, but they cannot fully understand that, and therefore we studied the microbiome of these sections, and we found out that the microbiome has a very specific reaction on this cold acclimation. And you can see here in the first picture, so if they are in a under normal condition, so they are close together and all these Arabidopsis ecotypes harbor more or less a similar community. But if we have these cold conditions, so then the network is more loose and we can identify that they react in another way, and we also find some bacterial genera which seems to be involved in this procedure. So this is an ongoing project, and we will try to isolate these Sphagnum and Buchnera strains and look if we can also find an experimental evidence that they are involved in this cold acclimation procedure. So the third microbiome is from medicinal plants. Here we had the hypothesis that they have a very rich metabolism, and we have seen that there is a huge impact of this metabolism on the composition of the plant. And this is also more a practical project, because here this is with Egypt and with the Sechem University and farms. And this is a very interesting project. I think this is one of the largest biodynamic farms of the world. And they produced a lot of organic vegetables for whole Europe, but also a lot of medicinal plants for us, because they have very good ingredients there. And here we studied the interaction of the metabolome and the microbiome. And we found that there is an interaction between the microbiome and the metabolome, and in this case we also could really isolate several strains which are able to change the metabolome of the medicinal plants, and which could enhance the bioactive ingredient. So we have done a lot of studies at this site, and here again we found for the 3 medicinal plants which we studied, calendula, chamomile and solanum, also very distinct networks for the plants, for the diazotophs. So, and now we come to the vegetable, to lettuce. And this was an EU project which I had together with a colleague. And here it was more basic research to understand the impact of several factors on the microbiome. And what we have done here is we compared healthy plants with diseased plants and found out that it is more or less connected with a strong microbiome shift. So there's a long ongoing research also on the mode of action of biocontrol agents, and now I think that the microbiome shift is really important for all these activities on the plants. And whenever you have a diseased plant, you see also a microbiome shift, and for sure there are supporters of the pathogens, but they are also antagonists. But by comparing a lot of plants and a lot of studies, so then you can identify those supporters of the pathogen, but also health indicators. And here, studying these networks more in detail, which is not possible to explain here, we found that there are several species which are only abundant and healthy lettuce plants. And maybe this is also an interesting idea to go in this direction to identify new biocontrol agents. So here, this is also a similar study then presented by Philip, but here for the lettuce. We used also a very unique collection from the Arche Noah. This is a facility in Austria, and they store seeds. And they also cultivate all these old cultivars. I don't know that before we start the project that lettuce is very old crop and used in the old China and Egypt and so on. And there exist very exotic lettuce cultivars. And here we studied 8 of them. And we identified a core microbiome. So all were in the field and all on the same site, so with the same soil. And we identified a core microbiome of 50%, but there was also a specific proportion of bacteria, and this means that domestication or breeding changed the microbiome, and now there is evidence for several crops, and there are several studies published showing this for maize and for other crops. So, but this is important if you think afterwards on our health. So if it is important if you eat this lettuce cultivar or another one, and yeah. So I started with my picture, if you remember, of the lettuce field, but also there were a picture with a confocal microscopy of the rhizosphere, which is then densely colonized by diverse microorganisms. And here in this project, we studied the phylosphere and also the rhizosphere, but for a long time we were not able to identify, to visualize the bacteria associated with the lettuce leaves. And it took really a long time, but then we realized that there is a wax layer, and this wax layer has an antimicrobial activity, and there are more or less no bacteria on this wax layer, but the bacteria are able to invade, and inside of the lettuce leaf we have a dense colonization of microorganisms and a high diversity of microorganisms, so which we eat. So, and interestingly, the dominant phylum were the enterobacteria, so this was first a surprise. But now for us themselves, we developed the hypothesis of natural vaccination by lettuce endophytes, because at the end it is, in most of the cases, very healthy. And I forgot to mention that we also studied the occurrence pattern of the leaves, and we found out that especially lettuce has a very loose network of bacteria, and it's very easy to invade into this system. And maybe this is the reason that we have so many outbreaks with lettuce. But on the other side, if you try to do biocontrol, it works so well with the lettuce system. So all these things, I think, it is very important to understand these networks and then also to exploit the knowledge for application. And this is really my last here. This is also a long-term research with KWS. This is sugar beet seed producing company, and together with them we develop products, biocontrol products, and one of the most important problems is Rhizoctonia solani in sugar beets. And we were really able to develop a good cocktail against Rhizoctonia, but we have the problem that each year the KWS want to work with other cultivars. Yeah, I have to hurry. And therefore, we studied seeds. And here we found out that we have, first of all the seeds are full of microbes. And here you have these changes from the rhizosphere to the soil. By the way, we have 1/3 of archaea, which they selected from the soil, which I found interesting. And here, a difference between the tolerance and the resistant strains. So I think I could show you that we have these specific networks. And it is also possible, so I have only a few things, to go from the other side, from the ecosystem to the molecule, and you can really exploit this knowledge to find new biocontrol, stress protection agents, but also bioactive compounds and enzymes. Here we developed a new method with bait plants to harvest also the microbial fraction from mosses, for example. I showed you that they harbor very interesting bacteria, and we were able to really to catch consortia against stress. And yeah, it is also possible then to develop other biocontrol agents here. This is one, Stenotrophomonas, protecting a lot of plants against abiotic stress by the production of spermidine and osmoprotectants. Or we developed a product for lettuce. I showed you these gaps in the knowledge microbiome, which works well. It is with a company, and now we also, with our Dutch colleagues, we start to control human pathogens, and this is, I think, one of the last things. So you also can exploit the compounds produced by the plant microbiome, and here we exploit the volatiles, and a new startup was established from our research by my young co-workers. And yeah, and this is my message. So not only to look on the plant microbiome, I think it's very interesting to see the microbiome connection, because we have evidence that they survive, especially the endophytes, and it's important for our gut health, and on the other side also for our indoor health, because we have microbes from the plants as well as from the humans, and due to the fact that we live a lot of our time indoors, it is very, very important. This is the vision of my group, to produce healthy food for our health, and here are several of my colleagues. So, yeah, thank you for your attention.